important because most of the intestinal hormones are secreted from the enteroendocrine cell. And also there are some neurohormones kind, serotonin also is secreted from the enteroendocrine cell. And last but not least is the enterocyte, which actually help in the nutrient absorption. So recent study already showed that the microbiota has some role in the intestinal barrier regulation and intestinal development. But how during this neonatal period microbial profile actually changes? And this microbial profile, due to this microbial profile changing, is there any role of this microbiota in intestinal epithelial development or not? That is not completely understood. So with this, the main goal of my project was the understanding the association and then is there any causal link between the gut microbiota and post epithelial development during this neonatal to adult transition. So generally we can't use the human model for this, so I use the mice model. So we used a C57BL6 mice model. So what we did, I started with postnatal 1 day, the very early day, that is the first day of the postnatal period, the first day after birth, and continued till 28 days. And in between, there are several time points. And first, we collected the cecum because the cecum is the most the part of the mice intestine where all the microbiota popularize. So most of the concentration of the microbiota is there in the cecum. So we analyze the cecal microbiota. So what we did, we first tried to see what is the total bacterial abundance. Total bacterial abundance, how it is changing during this neonatal period. So for that, we used a qPCR. We designed a probe against the 16S rRNA gene. So what is the 16S rRNA gene? So it is the conserved gene sequence present in the prokaryotic population, and it is used for their phylogenetic analysis. So we used a universal 16S rRNA primer to count how many total microbial population is there. And we can see there is a shift of the microbial total number postnatal 14-day onwards. So here you can see the significant changes is coming postnatal 14-day onwards, and then it reached a plateau. Then what we did, we wanted to see, yes, we checked the abundance, but what are the major composition of this microbiota during this period? So for that, we did a next generation sequencing analysis using the 16S RNA gene sequencing. And we found that in the very early days, the majority of the microbial population phylum is coming from the Proteobacteria phylum. So you can see most of the more than 60% is coming from the Proteobacteria. But suddenly, the interesting thing what we observed in the postnatal 14 day, a unique phylum called Vericomicrobia appeared at postnatal 14 days. Okay, and gradually their population decreased over 21 and 28 days. And when we see the what are the major population of the microbiota from 21 and 28 days, we found that bacteroids and firmicutes are coming. So bacteroids and firmicutes are more adult kind of microbiota. So as the ages increases, the adult microbiota population is localizing in the neonatal intestinal layer. Then we wanted to see how the diversity is actually changing. So diversity is how many different type of microbiota is there. So how many different kind of species is there? So for that, there are different kind of index we use. So one is the Shannon index. So what Shannon index do? It actually calculate how many different kind of bacterial species or OTUs are there. And also it sees whether they are evenly distributed or not evenly distributed. So there are two factors that they actually calculate. And when we analyze the data, we found that yes, it is changing from the postnatal 14-day onwards. The shift is coming. And further, we did a beta diversity analysis. So alpha diversity is within one population and beta diversity is between the population. We want to see how the microbial composition looks like. So for that, we did a principal component analysis to see how the microbial population actually clustered, whether they are clustered together or separately. And what we found interesting is that postnatal 1 and 7 day actually overlaps together, their microbial population. And the main divergence is actually coming from the postnatal 14 days. So here you can see the distance. The PC1 is 60.2% and the distance is also high. So they are totally diverged population. So divergence is coming from 14 day and then it is again changing. So we see that there are some changes in microbial population, but how the intestinal development is actually occurring in the host intestinal development. So for that, we used we wanted to see how the barrier integrity, that is the intestinal integrity, is actually developing. So for that, we actually used a in vivo gut permeability assay. So it is a functional assay of the barrier integrity. So I will just briefly tell you what we did. So there are actually one dye that is called 4-kD fixidextran. So what we did, we actually gavaged the mice with 4-kD fixidextran, and after 4 hours, we sacrificed the mice and collected their blood sample. So what actually will happen at that time? So that is the 4-kD fixation. So this is a dextran molecule attached with the fixid probe. So they will. So when we gavage, it will be present in the luminal site, and as the time passes, so it will come from luminal site to the blood vessel. Okay, so these are the paracellular permeability or paracellular junction. So from this will cross the intestinal epithelium and come to the blood vessel. So if our gut is more permeable, there will be more fixed extent in the blood vessel or in the systemic site. So that means the integrity is compromised. So here you can see when we plotted the serum fixation concentration, you can see that postnatal 1 day, there is the highest concentration. That means gut integrity is not actually developed at all. And there is a biphasic kind of nature of this drop. So first drop is coming from 7 and 40, and then again one drop is coming at 21 and 28 days. So we can see that by 28 days, the gut integrity is completely developed. We also then checked what are the major protein could be involved. So there are some tight junction protein that actually maintain the paracellular permeability. So during this time, the claudin 3 is the important one during the postnatal time. And we also seen that postnatal 14 day onwards, so you can say after 14 day, there is upregulation of this claudin 3 gene expression. And then what we check, as I already told, there are different kind of intestinal epithelial cells that actually help in maintaining the epithelial layer and barrier integrity. So we use different kind of markers, antibody markers for each type of cell. So first one is the Ki67. So it is a proliferation marker. And when we did a RT-qPCR cell analysis, and also we did a immunohistochemistry to check their protein expression in the ileum section. So what we found is that their population is actually increasing till 14 day, and after 14 days, suddenly there is a drop in their population. We also found the similar kind of trend in another marker, that is the chromogranin A. So chromogranin A marks for the enteroendocrine cells that I already discussed. So there also we found a similar trend. And from here we can say that there is a decrease in specific cell population from postnatal 14 day onwards. So maybe the 14 day is a major time point, because we also observe there is the shift is coming in the microbial population from the postnatal 14 day, and also in the epithelial cell markers are coming from the major change is coming from the postnatal 14 day. Then what we check, because we check that there are up regulation of the varicomicrobial population, and from varicomicrobial phylum there is one particular genus is there that is called Akkermansia. So these are the mucus degrading bacteria. So we check that this Akkermansia population is also high at P14 day, and previous literature also showed that this Akkermansia actually degrade the mucin layer. So we are also interested to see how the mucin gene expression is changing or not. So we found that the mucin gene expression is actually low at the postnatal 14 days, because there are a higher number of the Akkermansia at that time point. We also check the goblet cell that actually produce the mucin or the mucin protein. So we did a Alcian blue patch staining to particularly stain the goblet cell in the intestine. And we also found the similar trend that after 14 day onwards, there is an increase in their population. The goblet cell population is increasing after 14 day onwards. So then we did a correlation matrix analysis to see whether there is a correlation between the microbial phylum and major intestinal development parameters that we observe. And we found that yes, the proteobacteria is actually negatively regulated, negatively associated with the markers, and bacteroids and firmicutes are positively regulated. So bacteroids are positively involved with the claudin 3 marker, and varicomicrobial is positively associated with the proliferation marker. So from here we can see, yes, there could be an association with the microbial development and intestinal epithelial development, and postnatal 14 day could be an important point. So, but this is an association study. So from here we cannot say whether this microbiota has any role or not. So what is the cause and what is the effect? We cannot say. So for that, we have to shift to the cause and effect study. So we move to the causation. So for causation, I would like to say, for to prove any kind of causation during gene, what we do actually, we knock out the gene and prove their function is actually hampered or not. So similar kind of thing is also there in microbial studies or microbiome sciences. So ideally, we have to eradicate all the microbiomes. So for that, people use the germ-free model. But you know that the germ-free model is actually not developed in India at all because various reasons. So there are other methods to modulate the microbiota or deplete the microbiota. So people use the antibiotics to perturb the microbiota. So then our question is, what is the effect of the intestinal microbiota perturbation on postnatal gut epithelial development? So previously, using different kind of antibiotics, we already showed that antibiotic could have effect on this microbial population, and it also depend on the different classes of antibiotics. Okay. So now the major goal of now is that we wanted to perturb the gut microbiota development during the postnatal development period and wanted to see this factor, whether it can cause a developmental error in the intestinal epithelium, is there is any kind of age dependent role of particular gut microbiota, and third was whether a diverse population of microbiota is needed or only one phylum can do the needful. So my goal is to completely eradicate or deplete the microbial population early from the neonatal days. So for that, we used a ABNM cocktail. So it consists of ampicillin, vancomycin, neomycin, metronidazole. So they used a broad spectrum antibiotic that will target the anaerobe and aerobe and also gram-positive and gram-negative bacteria. So it is a well-used antibiotic cocktail to eradicate the microbial population. So what I did, I started with the postnatal 3-day because it is very difficult to manipulate or gavage any kind of thing during the postnatal 1 day. So I started with the postnatal 3-day and I did a daily gavage of this antibiotic cocktail to the neonatal mice. And in the control group, we gavage the PBS. And in between this time point, that is the 7-day, 14-day, 21-day, and 28-day, we sacrifice the mice. And this is also an extended plan where you can see I actually pool the mice. So in sciences, biological sciences, we use the term biological replicates. How many biological replicates you are using, 3 or 6. But here the biological replicate concept is somehow different because we have to pool mice, because with one neutral mice, we don't get enough biosamples. So what we did, we pulled 2 to 4 pups from each litter and used as a biological replicate. So here you can see these are the dams from different cages. So we do a breeding in different cages. So their litters are there. And from this litter, maybe we took 2 to 4 pups, we pulled their sample and considered as a one biological replicate. So it will be the first, it will be the second, it will be the third biological replicate. And also we took my sample from different cages because there is a cage variation thing in the microbiome study. So our first objective was whether the antibiotic regime that we use is actually doing the perturbation or not. So what we found interesting is that the microbial population is actually depleted at only postnatal 14 days. So you can see that postnatal 7 days, there is no difference between the two groups. ABNM is actually effective at postnatal 14 days. And interestingly, after postnatal 14 days, their population is again increasing. The total abundance of the microbiome, total bacterial population is increasing. So that's why we checked to see, we're interested to see how the diversity is. But what we found interesting that at postnatal 14 day, there is no significant change in the diversity. Whereas the significant change in the diversity is actually in the postnatal 21 and 28 days. When we also see the beta diversity plot, PCA plot, we can see that the postnatal 14-day population is not that much diverse compared to the 21 and 28 days. So we thought, what could be the reason? So we hypothesized that maybe one phylum is getting actually overpopulated at that time point. That's why the diversity is actually changing. So for that, we look into the phylum level, and here is the result. Here you can see that postnatal 21 day and 8 day AVNM population is mostly comprised of the proteobacteria. So more than 95% of the microbial population is from proteobacteria. So their diversity is less. So that's why the alpha diversity is changing. But in the postnatal 14 day, you can see although proteobacteria is there, but there are other microbial population. So what we hypothesize that during this point, what is actually happening, maybe the proteobacteria phylum is actually getting an antibiotic resistance and they are getting overpopulated and their population is increasing at 21 and 28 days. So next what we did, we collected the information from the top genuses, top 10 genuses and their corresponding bacterial phyla. And what we found that at postnatal 14 day, there is a significant difference in the Akkermansia population. And this Akkermansia is coming from the Vericomicrobial phyla. Then further, we actually validate the Akkermansia population because this is a NGS study. So we have to validate it. So we did a qPCR based analysis with particular gene sequence targeting Akkermansia. And we found that yes, their total absolute abundance is also decreasing in the AVNM group. So from here, our findings actually indicate the postnatal AVM treatment could drastically alter the fecal microbial diversity and shift the composition to a proteobacteria enriched gut ecosystem. And another thing is the postnatal 14 day could be an important period where the Akkermansia population is high. Now we see the antibiotic can modulate the microbial population, but whether this antibiotic mediated microbial depletion or perturbation has any effect on functional barrier integrity or not, that we have to check. So for that, again, we did the PIPC analysis that I previously described. And here also we found that the postnatal 14 day it is showing the major changes. So it is dextran concentration highest in the postnatal 14 day AVN group. That means the gut barrier integrity is compromised at 14 days only. And when we check different kind of tight junction marker, also we found the same pattern like Claudin 3, then Zonocludin 1. All their gene expression is downregulated at postnatal 14 days. And after 14 days, they show a pattern of recovery. So they are actually trying to recover their gene expression values. Further, we validate with our immunohistochemistry study that whether the population or the Claudin 3 marker is actually less or not. And we found that yes, in the antibody level also, they are actually less in the Claudin 3 with the mean fluorescence intensity value. Next, we checked for the other marker that I already discussed, that is the Ki67 and chromogranin. Both these markers also showed a similar pattern. That is a postnatal 14 day, they are majorly depleted. Their cell population majorly depleted at only postnatal 14 day, and then they are showing a recovery or resilience. So you can see that Ki67 control group, their population is there, but AVNM it is actually depleted. Similar pattern was also observed from the lysozyme 1, that is the panic cell marker. So from here, our data suggests there is a age dependency effect of the antibiotic treatment, where the barrier function and intestinal epithelial population was relatively high at the postnatal 14 day. And after the postnatal 14 day, maybe there are some mechanisms that hosts try to recover the detrimental effect of the microbiota population during this sustained antibiotic pressure. So we found that the postnatal 14 day could be an important time point for the intestinal development or neural development. So we checked because the barrier integrity is compromised at the postnatal 14 day. So we checked whether the systemic inflammatory cytokines are also changing, because if there are some breaching in the intestinal epithelial layer, microbial products will come to the systemic side and it could lead to a systemic inflammation. So we check two different markers using ELISA. One is the TNF, one is the IL-6. So these are the pro-inflammatory markers. And we found that in the AVNM group, their concentration is high in the serum. We also check the serum LPS concentration. So the LPS is the outer membrane of the bacteria, gram-negative bacteria, you know. So their abundance is also increasing in the postnatal 14 day, because we showed that there is a enrichment of the proteobacteria, and proteobacteria are mostly gram-negative bacteria. So this LPS layer is LPS population is also coming, concentration is also increasing at that time point. So from here we draw a predictive mechanism. What could be the scenario in the intestinal gut at the 14 day? So due to our AVNM treatment, there is an imbalance between proteobacteria and vericomicrobial population. So proteobacteria is high and vericomicrobial is low. So there is a microbial dysbiosis. And this could lead to a barrier integrity disruption that I showed from the Fitz and different tight junction marker. And due to this barrier junction disruption, there is a breaching of the microbial products. So microbial products are coming from the luminal side to the systemic side. So the LPS concentration is high, and it could lead to the concentration of the inflammatory cytokines like TNF and IL-6. So from here we can see that the epithelial layer actually hampered. But this microbiota also crossed up with the different kind of mucosal immune cells also. So there is always a crosstalk between mucosal. This microbiota actually educate the mucosal immunity. So we want to dig some more further. So we did a RNA-seq analysis on the mucosal layer. So we took the postnatal 14-day ileum and we did a bulk RNA-seq from the intestinal epithelium. And we found that the transcriptomic makeup is totally different between control and AVNM group. So you can see the PC1 axis is 76.7% and their total diverged population. Then we do a volcano plot analysis to see what are the top differentially expressed genes during our antibiotic treatment. So we found these genes are actually downregulated, significantly downregulated. And when we search for their function of these genes, they're actually indicating some role of the immune cells, like immune mechanism or kind of thing, like B cell activation, then antibacterial immune response, then macrophage activation. Mediated gut microbiome perturbation, immune-related function are also getting modified or dysregulated. So from here, next what we did from our RNA-seq data, we do a targeted gene set analysis because we know from our study that the Paneth cell population is actually depleting, and Paneth cell has enormous role in antibacterial peptide population production and innate immunity. So we tried to find all the markers or all the genes that are actually expressed in this cell, and we found that different genes like REC3B, then CSF1 are actually significantly downregulated in the AVNM group. Next, what we did a function enrichment analysis. So for that, first we used a cake databases, and what we found from this, we used the downregulated gene set in our AVNM group, and from their function analysis, we found the top two functions are, one is the intestinal immune network for IgA production, and second one is cell adhesion molecule. So IgA is also important because IgA actually regulates the barrier integrity and it can also modulate the microbial population. So we wanted to validate the RNA-seq data. So what we did, we checked the cecal IgA concentration. IgA concentration in the cecal samples. And we found that yes, they're actually significantly less in the AVNM group. And also we found the cell adhesion molecules like tight junction protein integrins are also downregulated. And when we tried to search in the another database, that is the gene ontology biological process, there also we found that immune-related function like adaptive immune response, positive regulation immune response are downregulated. So from here we can say that this can alter barrier and intestinal function. So there are some crosstalk in this barrier integrity and immune cell function. So we showed that the postnatal 14 days the important period and microbiota division at the time point has some detrimental effect. But whether if we can try to repopulate with the healthy microbiota that we told, whether it could be transferable. So that would be the actual evidence of our causal study. So what we did, we tried to transplant the microbiota from control group. So we planned an experiment that is called cecal microbiota transplantation. So what we did, so here is the snapshot of the study design. So there are three groups. One is the PBS group where we did nothing. We just supplied the PBS. One is the AVNM plus PBS group. So here what we did, we perturbed the microbiota till 13 days using the AVNM. And after that, we gavaged with the PBS. And in the third group, where we actually transplanted the microbiota. So what we did, we perturbed the microbiota with AVNM. And after that, postnatal 14 day to 16 day, we gavaged with the control microbiota population. So we have two microbiota cecal samples from AVNM group and control group. So we took out the control microbial population of cecal sample and gavaged the AVNM perturbed mice to see whether this microbiota can again recolonize there or not. So for that, we used the abundance to see whether the gene copies is actually increasing or not. and we found that the total abundance of the microbiota is actually getting recovered to 86%. So compared to the previous group. So in the evening group, it is 67%, and again it is increased in the 86% to the ABN plus CMT group. So there is a recovery of the microbial population. Next, we wanted to see whether, because this microbiota is not present in the fecal sample only, because microbiota is present in the mucosal layer also. So we wanted to see the mucosal attached microbiota is also recovered or not. So for that, we planned a study called fluorescence in situ hybridization. So we tagged a probe that is called EUV338. So it is a 16S RNA probe tagged with a specific Cy3 dye. And after tagging, we do a in situ hybridization study. So from here, you can see these blue color daphy are the host epithelial cell, and these are the microbial population. So you can see in the previous group, there are enormous bacteria present attached to the mucosal layer. In the ABN plus previous group, they're actually depleted. And again, in the CMT group, they are recolonizing, but not to the extent of the previous group. When we checked for the diversity, whether their diversity actually increasing or not, we found that the diversity is not completely destroyed, they're partially destroyed. So here you can see there is no significant difference between the PBS and CMT group. Also, when you can see the beta diversity, the PBS, the CMT group is actually deciding between two groups, PBS group and ABN plus PBS group. So from here we can say that their diversity is actually partially destroyed, not completely destroyed. So what could be the reason behind the partial restoration? So we did again the phylum level analysis, and we found that in the control group there are majorly three phyla: Vericomycobia, Bacteroides, and Firmicutes. Whereas due to perturbation, the majority of is coming the Proteobacteria. But when we try to restore these two phyla, that is the Bacteroides and Firmicutes are actually not getting restored, whereas only the Vericomycobia phylum is significantly getting restored. So when we do a genus level analysis, also we can see that this Vericomycobia, that is Akkermansia, is getting restored, and other microbial genuses are not getting restored. Further, we validated with our qPCR study that yes, their actual abundance is actually getting restored. Vericomycobia abundance or Akkermansia abundance is actually getting restored. So now we can say that the microbiota population, there is a selective restoration of the microbial population. So whether this selective restoration of the microbial population can modulate the barrier function that we observe, that detrimental effect, can it restore the detrimental effect or not? So for that we did again the fixed dextrin analysis, and we found that the barrier integrity is actually increasing. So here you can see double y-axis plot. So one is the serum fixed dextrin concentration is actually getting depleted, and barrier integrity is getting restored. When we check for the different marker like claudin 3, that means that tight junction marker is getting restored. Also the serum LPS level is coming to the baseline level. So that means the barrier function is getting restored. And also the other epithelial cell markers are also getting restored, like Ki67, enteroendocrine, lysozyme 1, all the cellular population are also getting restored due to the same treatment. So from here, we can summarize. So if you can see, it is a road, and microbiota is regulating the host intestinal epithelial development. So postnatal 14 days is the crucial time point. We proved that. So why it is crucial? Because that time point, any effect in the microbial population could be harmful in intestinal epithelial development. And if we see what is the major reason behind that, the Vericomycobia could be majorly behind that. So it can restore the barrier integrity, intestinal epithelial cell population, and also restore the serum LPS concentration. So we can say that the diversity is not that much important. So maybe one particular bacterial phyla, that is the Vericomycobia or signature microbial population, can restore or has the effect on the intestinal development. So now what's next? So we know that this microbiota has role on that, and nowadays researchers also showed this microbiota actually produce enormous number of metabolites. So what are these metabolites? They are actually small molecules. When this microbiota decompose the fiber-enriched food in your colon, they produce enormous number of these small metabolites, and they could be different kind of in their chemical structure, from amino acid, vitamins, then short chain fatty acid. etc. And nowadays literature also showed that this microbiota derived short chain fatty acid has role in tissue barrier function. Also, it can modulate the stemness of the intestine. So people showed that the derived metabolites or microbial metabolites has some role. But what is the significance in our study? So we showed that the microbiota is the cost and post epithelium is the effect, but how it is doing. So we thought maybe the metabolites are doing. So for that, we planned our experiment, future experiment in a different way. Because what is happening in the in vivo system, it is very difficult to say any kind of direct effect because there are so many factors like there are so many microorganisms out there. Also, there are some confounding factors like immune cells. They can also modulate the host epithelium. So we generate a ex vivo model that is a 3D organoid model. So only the epithelial cell layers will be there. Okay, and we wanted to see whether this metabolite has any role on it. So for that, our extended plan was we have a part of that microbiota at postnatal 14 days. So we collected their cecal samples. Here we have the cecal metabolites, and we treated with the organoid to see whether this metabolite has any direct effect in epithelial proliferation or differentiation on. So in our experimental approach, what we did, we isolate the microbial metabolites on two groups, that is the control and part of mice. First, we characterize the microbial metabolite using LCMS, and then we validate the metabolite function using an ex vivo organoid model. So before coming to organoid, I know that is a new topic. So I want to give a brief history. So the organoid was first developed by Hans Keller. So he's a scientist from the Netherlands. So they actually first developed the intestinal organoid. So there are some concepts because cell line is a 2D cell culture. So there are monolayer. Then other complexity come with the transwell, where you co-culture two different kind of cell with some kind of cell culture insert. Then comes spheroid and then come organoid. So there are some difference and important difference between spheroid and organoid because most of the cancer study they use spheroid. But what are the different major difference in the spheroid and organoid? The spheroid are unicellular because there are one type of cell in the spheroid, and organoid has different kind of cell. Spheroid can be developed by culturing one cells, so one cell attached with each other and can develop the spheroid. But for organoid, you need the stem cells. Without stem cell, the organoid cannot be developed. So these are the main reason. And what are the major advantage of the organoid? Nowadays, organoid are used for the drug testing, then different kind of modeling disease, intestinal diseases, post microbiome interaction that I am interested in, and also biobanking. You can see that during the COVID time also there are various people use this organoid model for biobanking and different kind of drug testing. And what are the major feature of the organoid? So it can differentiate it to multiple lineages of the cell. So from stem cell, it can differentiate to any kind of cell. It is a self-renewable, so you don't need to again and again do a cell culture kind of thing. It can recapture the organ architecture. So in the cell culture, there is no organ architecture maintained. So if we are saying the intestine, there is a lumen, there is a polarity of the cell. All things are not recaptured in the cell culture, but in the organoid, you can see there is a lumen and there is a polarity of the cells. So there are different approach of the organoid development. One is from the adult stem cell. So you can use animals or some patient derived also you can use. And another is the iPSC, that is an induced pluripotent stem cell. Also, you can develop the organoid. So for my study, I used an adult stem cell because in the intestine, I have already showed that there are some crypts are present. So in the crypt, there are some cells called LGR5 positive stem cells. So these are the adult stem cells. So from there, you can actually generate the organ. So what I did, I collected the mouse ileum part. Then we collected the crypt portion, the intestinal crypt, which considers that LGR5 positive stem cells. And this stem cell cannot grow without giving any kind of thing, any kind of transcription factor. So you have to provide some kind of transcription factor. So there are some specific transcription factor, and you have to provide matrigel because the matrigel acts as an extracellular matrix. So when you provide both these factors, it can differentiate into the all the cell layers that are present in the intestine and produce the organoids. And these organoids can be developed for various organs from liver, kidney, lung, stomach, brain, etc. So also I want to show you that recent trends in the organoid. So you can see that from 2015 there is a huge increase in use of the organoid, and during COVID you can see there is a spike in the organoid using. And also you can see there are publication trend using intestinal organoid for the gut microbiota and metabolite study. So if you want to visualize the organoid, here is the organoid, how it looked like under the microscope. So you can see that day one. So this is basically the tips of the intestine. So they are not actually developed to organoid. As the day increases, we can see there are some budding structures. So this budding structure represents the tips of the organoid. Then you can see a darkening of the lumen. That means the lumen is actually getting generated. And by day 7, you can see a mature organoid. You can also see how the surface area of the organoid actually changes, significantly increase by day 7. So when we use this organoid and we use the sickle supernatant to treat it, we found that there is a difference between control and ABN. So we have two groups, control and ABN, sickle samples from postnatal 14-day mice. We collected the sickle supernatant that contain the metabolite, and when we treat the organoid with the metabolite, we found that there is no change in the control growth, but there is a huge change in the ABN growth. So you can see rarely can see a budding structure there because they are not actually bud. That means their proliferation is hampered. So all these factors are there. I am actually not presenting all my data here because these are not published. You can also see different kind of organoid here also, like MUC2, different kind of cellular layers like MUC2 are getting the fluorescence from the MUC2 lysozyme claudin 3. Also, what we did, we also developed a confocal microscopy protocol to image this organoid. So we can also do a 3D study with this organoid, and you can see a 3D image of this organoid here. So from here, you can say that I used a multi-omic study comprised of metagenomic, transcriptomic, and metabolomic to find what is the role of this microbiome. So we found that the postnatal 14 is the important time point for the microbiome development, where this microbiome development and the varico microbiome is the key player. Also, these microbial metabolites has role in the organoid development or organoid proliferation and etc. So whether this study has some translational role or not, because this you can say that this is a basic study, but no. So there has some translational future. So we found that the Akkermansia is the important microbiome present in the neonatal tank farm. So it could be used potential probiotic because there are no specific neonatal probiotics we know. Also, we can use these metabolites as a postbiotic treatment. So if we found some specific metabolite produced by this microbiota, we can use these metabolites as a treatment during the neonatal point. We can eradicate the microbial population. We will not supply the probiotics. Rather we treat with the postbiotics. Also, we can use different kind of co-culture models to generate some complex kind of organoid with the immune cells, whether these immune cells has any role or not. So with this, I want to end my talk. So I'm very much thankful for my PhD supervisor, Professor Paulokai, to give me the independence to do all the work. Also, our lab, different kind of facility that I used from CIS, then Central Instrumental Facility, all the travel support and all. So with this, thank you. And I was also involved during the few days with my PhD project with a company called Microbiotics. So this company is developed from our lab only. Outside actually developed this company. So this is the first startup coming from NISER. So we are actually developing two products. One is the gut function test and one is the personal probiotic. So you can also reach out to them regarding any kind of information. So thank you.